Hello, everybody. Thank you all for joining in today for the Bioinformatics virtual seminar. It's my pleasure to introduce our speaker, Dr. Daniel Rowe. Dr. Rowe is currently a Chief of High Performance Computing at the Laboratory of Computational Biology, the National Heart, Lung, and Blood Institute, NIH. He got his PhD in chemistry from Stony Brook University in 2007, working with Professor Carlos Simmerling. Dr. Rowe's distinguished career includes significant postdoctoral research at NIST with Dr. Anna Chaka for 2 years and Rogers University with Professor David Case for 2 years and 4 years of staff scientist at the University of Utah with Professor Tom Chittam before moving to NIH in 2016. He is a long developer of Amber and Charm. Amber and Charm are a suite of programs that allows users to carry out molecular dynamic simulations, particularly on biomolecules. He is a member of the Amber Executive Committee since 2023, helping to direct the future development of Amber. Um, Dr. Rowe has over 60 publications ranging from various topics in biomolecular simulation. And not to miss, Dr. Rowe is the lead author of CPPTRAJ, a fast parallelized program for analyzing the simulation data from various AMD software packages. And CPPTRAJ is the main program in Amber for processing the coordinate trajectories and data files. This work alone has received, I believe, over 7,000 citations. Today, he will be talking with us on the recent advancements in the molecular dynamic simulation, trajectory analysis using CPPTRAJ. And Dr. Rowe, we are all excited to hear from you. Everybody join me in welcoming Dr. Rowe. Take it away, Dr. Rowe.
Okay. Hello. Thank you. Thank you, Rajan, for that introduction and for inviting me. Um, so the title, Recent Advancements in um, Molecular Dynamics Trajectory Analysis, right? So I was actually telling Rajan before everyone hopped on that when I gave this title, I didn't realize that the talks were supposed to be 40 minutes long. I thought it was more like a 25-minute talk. So um, so I read the email again. I was like, oh, wait, 40 minutes. I'm going to need to talk about a little bit more. So actually, I'm going to get into a bit of the why for CPPTRAJ as well as the what. And I think it's a better story overall. So um, yeah, but about me. So Rajan mentioned that I'm now Chief of High Performance Computing. I'm running the Lobos cluster at the NIH, which is the second largest cluster at the NIH after BioWolf. Um, I sort of it's been taking up most of my time since January. So unfortunately, I haven't had as much time for research. I'm hoping to get back to that soon. But I wear a lot of hats where I am at the NIH. So that's just one of the things I do. Software development, Amber, Charm, CPPTRAJ, that's still ongoing. I've done some methods development stuff. So here, these are just all like a couple of publications, recent publications over the past couple of years, and then basic research. So again, especially the basic research and methods development, I haven't had as much time for, which is why the recent developments, really recent means like over the past couple of years, but I still think it'll be interesting. Um, and so more than a couple of years ago, actually, I'll be going back a lot farther in time because I think the development of CPPTRAJ really comes naturally out of the quest for more sampling. So I'll get into that, the development of CPPTRAJ as a fast analysis tool. Then I will get into some recent advances in CPPTRAJ, and then a topic that I thought would be interesting because I've seen a lot of people, especially newer students trying this using ChatGPT for creating analysis scripts. I wanted to delve into that a little bit. It's actually very interesting. and then depending on how much time, um, maybe some advice useful. Um, that depends. So anyway, so now going back a long time ago, about gosh, was it 24 years ago? My goodness. So I joined the Simmerling Lab as a grad student in 2001. There I am on the left, fresh-faced, full of promise and hope. 2 years later, on the right, 2006, beaten down, broken, bearded. Grad school could be really tough at times, but it wasn't that bad. It was pretty fun actually, and I learned a lot. Um, but back then, MD was much, much slower. So these are some Amber 9 benchmarks from about 20 years ago that I managed to sort of scrape together out of the like archives of the Amber web page. Um, and anyone who's done any sort of MD simulations over the past 10 years, you're probably used to seeing timings in nanoseconds per day. So note that these timings are in picoseconds per day. And this is the Factor 9 benchmark. It's about 90,000 atoms. It was a pretty big system back then. And we top out at about, for PMMD, across 128 of these IBM Power 4 nodes, at about 7 nanoseconds a day. So just for comparison, on a modern H100 NVIDIA GPU, you can get about 357 nanoseconds a day on this benchmark. That's about a 50 times increase. Um, so back then, MD was just much, much slower. And turns out, as a consequence of that, an early lesson that I learned was that independent simulations often didn't match. So this was one of the early systems I worked on. It's a three-stranded beta sheet model, DPDP, that it's kind of cool. It's got, you can see the structure over on the right. It's got these two D-prolines in the hairpin turns that sort of lock those turns and help the sheet form. But so I did some straight MD simulations on this system. One on the left starting from the native folded structure, one on the right starting from sort of a linear structure, and they never agreed. They never met. And this is now each of these is over 200 nanoseconds of simulation. This was months of simulation time back then, and just couldn't get it to agree. Um, and so why is it important for simulations to match? Well, accurate modeling of any phenomenon requires the observation of multiple events. This is sampling, right? And when you do any sort of science experiment, you repeat, over and over. You want to see the result. You want to get some sort of statistics on it. And so results from independent simulations should agree. So if I start at different areas on the free energy landscape, then the simulations should start to look similar. They should they should adopt similar conformations. They should converge, right? The structure population should agree. If you don't have enough sampling, then you're going to have poor convergence. And if you don't have convergence, then your structure populations are not Boltzmann weighted, and you can't do things like calculate thermodynamic properties like free energies, which is something that we often want to do. So convergence is difficult to achieve because it's difficult to do enough sampling. And an early example, a thought experiment I did as a grad student was I thought about a six-sided die, right? It's a very simple system. It's got six possible outcomes, and you know intuitively that each outcome has a probability of 1/6. So how many rolls, so say you're just rolling this dice and you're recording the result, how many rolls before each number has a probability of coming up 1/6? So I just wrote a C program that just called RAND, and the results surprised me a little bit. So the probability for each side coming up, if you look, even after 100 tries, none of it's really converged onto 1/6. Six is pretty close. It actually took out until like 20,000, 30,000 tries before those curves settled down to that 1/6. So it's this result. It's intuitive that the more sampling you can do, the more converged your results will be. But what surprised me was that for such a simple system, it took so much sampling. And so imagine now for doing molecular dynamics on a system that's got like 100,000 atoms and that many more degrees of freedom, and you can imagine that that's why convergence can be really difficult. And so in terms of sampling, I like to think of this as just have you covered enough ground. And so um, This is sort of a map of when, European explorers were venturing sort of across the Atlantic Ocean and trying to circumnavigate the globe. And, here we know what the world looks like, nowadays. We have GPS, we have satellite imaging. But back then it was basically like they were on ships and they just kind of looked and, made measurements and were like, it kind of looks like this, right? So here's a map from about that time, it's 1544. And the shape, the world is kind of recognizable, like you can sort of recognize the continents, but there are some problems with this map, right? First, there's incomplete sampling, right? The North American continent just sort of disappears off into nothing, and like Australia is not a thing, right? It's just not on this map. There are also issues with the things that are there. There's poor accuracy, right? There's, the relative sizes of like the Red Sea, the Caspian Sea, the Indian subcontinent. They're 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 not quite right. They're not what we know they are today. And one of the problems is that you can't even ascertain accuracy issues in the areas with no sampling, right? So problems with accuracy, like there might be accuracy problems over near, Australia, but we don't know because there's no sampling there. So what can we do? Well, there's some tricks like enhanced sampling methods. Probably one of the most popular is replica exchange molecular dynamics, and that was still fairly new when I was a grad student. In 1999, Yuji Sugita and Yuko Okamoto um, came out with this paper. And incidentally, if you get a chance to meet Yuji Sugita, he's super, super nice, really cool guy. And we were implementing this method in Amber around the time that I was a grad student. Um, it's a conceptually simple idea where instead of one copy of the system, you have multiple replicas, and every now and then these replicas exchange information, either Hamiltonian information or coordinates, however it's done. There's information exchange, and this enhances the sampling. So again, going back to my original, simulations of DPDP, um, this is limited sampling with MD, poor convergence, apply replica exchange to it, and now with just 100 nanoseconds per simulation, per replica, now I've got starting from three independent areas on the free energy landscape, and now I've got um, pretty good agreement. I'm starting to sample all the same sorts of structures, and this actually led to my first paper, which in 2005, which was really nice because before this I had two just outright rejected. And it was brutal. I wasn't sure I was going to make it at that point. But with more sampling, that means there's more data. And so one of the big like advancements in the molecular dynamics field um, was the introduction of GPUs. And so you can see, so here are some benchmarks. This top graph shows some benchmarks for Amber, and the black line is the CPU benchmarks. It's kind of like what I was showing you guys before. And then the orange line is when the GPU is introduced around Amber 12. And now you can see these numbers just shoot up. And this led to an explosion of data because you went from taking, days to get a nanosecond to nanoseconds per day. Um, Concurrently, you get from megabytes per simulation to needing hundreds of gigabytes per simulation. And if you're using something like a replica exchange with multiple replicas, you can multiply that number even further. And so now there's this data explosion. And it's something that we wrote about when I was with Tom um, at the University of Utah. And that was, still, and GPUs are just getting faster. And so this problem is still here. And so now part two is about developing these faster analysis tools. And I want to show this slide primarily as an example of like things to not name your software. Like really just think before you name your software. So the history of analysis tools in Amber, the very first one was named Anal. And I don't know if they just never said it out loud or they just never like had a discussion about this, but like it's just really unfortunately named. Written in Fortran, it was quickly sort of succeeded by a program called MDNL. Again, poor choice of name. Written actually by Wilfred Van Gunstrin, who you guys probably know more as the guy who developed Gromos, but he did spend a year with Peter Coleman, and this program came out. I'm not trying to like bag on anyone's code. Like writing code is hard, but I was looking up. I was trying to get some history on all these things, and I came across this quote from Dr. Chris Edge on CCL, which unfortunately CCL is going away, the computational chemistry mailing list. But he called it a nightmare to use, and I was like, oh man. I mean, I'm sure people say that about CPPTRAJ, but I haven't seen that yet. And then also in the one of the official Amber publications in 1995, it was stated that the code was declared impossible to understand. So it needed some kind of successor. That successor, again, named Carnal. Again, terrible name choice. I don't know what it was with Amber analysis and bad names, but after that, finally, with a non-ridiculous name came PTRAJ, developed by Tom Cheatham. Um, Written in C, a little bit of Fortran. There's a picture of the late great Peter Coleman and Tom Cheatham. Tom basically still looks like this. He's like he refuses to
Mhm.
age. Um, and PTRAJ was great, and it was around. That was the program when I was a grad student. Um, and it was great. It had a lot more analysis than had come before, a lot more things that it could do. That's sort of a general trend, starting from the original analysis program with just like seven sort of different types of analysis. PTRAJ could do, 17 plus. So the scope of functionality was increasing, but not the speed. And that was a problem once the data explosion started to happen. And so enter CPPTRAJ. Very, I'm not good with names either, but at least it wasn't ridiculous. It's just C++ traj. It started as a rewrite of ptraj in C++. I was just teaching myself C++, ended up being Amber's analysis program, and it's been on GitHub since 2015 because I'd like to encourage, developers. And if anyone listening is like a C++ developer, you want to jump in, help out, I would love that. That I would encourage that. I've tried to make it very sort of software agnostic. It supports, Amber, Charm, NAMD, GROMACS, OpenMM, Desmond, a whole bunch of different MD packages, over 200 commands at this point. There are some Python hooks via a project called PyTraj by Hai Nguyen on GitHub. And importantly, for the stuff that I've just been talking about, it has multiple levels of parallelism since 2014 across trajectory parallel and for time-consuming analysis, OpenMP and CUDA. And so there was a real need for this, um, and as I'll get into. But so the parallelism in CPP-TREG is it's conceptually very simple. Say you have 1,000 frame trajectory, you have 5 processes, just the total number of trajectory frames are just divided up among all the MPI processes. So process 0, in this case, we get frames 1 to 200. Process 1 gets frames 201 to 400, and so on. And then each process, if you've got extra cores or you have, say, like a GPU available, each process can make use of OpenMP or CUDA to further accelerate actions. And again, there was a real need for this. So when I was at the University of Utah with Tom, we had access to a supercomputer called Blue Waters, which was awesome. Had over 20,000 CPUs, over 4,000 GPUs, and it really enabled us to do these really gigantic, like, hundreds of replica exchange jobs. But like, again, that generated a ton of data. And it was taking us hours and hours just to do a single process in serial. And so that's really what spurred on this development of a parallel analysis program. And so, we were able to get the the time taken to process Hamiltonian replica exchange um, trajectory, a series of trajectories down from 7 hours to 1 hour. And I mean, you could get it down even further by throwing more and more cores at it. So like the speed up was real, and it really enabled us to do some cool analysis. Um, and so now I'll get into a couple of recent advances in CPPTRAJ. This is not an exhaustive list. It's just sort of the ones that um, I thought were kind of neat. But you can always go to the CPPTRAJ GitHub page and sort of look at the recent pull requests. I did try to keep up with the like what's new in CPPTRAJ. Every now and then I update it, but I'm really behind. But anyway, one of the cool things that I did was to put the radial distribution function calculation on GPUs. So it can be a really intensive calculation. You need to calculate a lot of distances, right? For a radial distribution function, you're doing, say, if you've just got 15,000 waters, you need over 100 million distances per frame. And so if you've got tons of frames, that can take forever. But so over here on the right, you can see that now we're going from taking hours to process and calculate a radial distribution function. With OpenMP, it scales very well. With MPI, you can scale even beyond that. And then CUDA just makes it that much faster. And you can have CUDA and MPI together at if you have multiple, in this case, I'm using multiple V100 GPUs. You can really get the time down to from minutes to even seconds. So that was good. Another one was parallelizing the grid inhomogeneous solvation theory calculation. And so that's GIST. And it just is a cool technique that lets you determine essentially like how well water is going to bind to different areas on your solute. And so it's made up of two calculations, the solvation energy and the first order solvation entropy. The energy time scales with the number of atoms. There were actually a couple of efforts made already to accelerate that, putting it on GPU, adding particle mesh ewald, both of which did accelerate things, but the GPU had no PME, and OpenMP actually scaled really poorly for PME, just for technical reasons, the reciprocal space calculation hasn't been well parallelized yet. And so my idea was to use MPI to just do across frame parallelization for the energy calculation, and that it works in conjunction with the GPU or the OpenMP or PME. And you can see on the right, again, where calculations that are taking, over 1,000 seconds now take on the order of like, 20 seconds. And there was an additional wrinkle, though, with the GIST calculation. So there's not just the solvation energy, there is also the entropy. And the entropy calculation is different because it's not independent. Okay, each frame depends on other frames, right? It depends on where the water has been to do the entropy calculation. And I was finding out that even for a small system, like a 7,000 atoms system, um, the entropy calculation was scaling not with the number of atoms, but with the number of frames. And so with the entropy calculation in serial, it was taking over 1,000 seconds, and 88% of that was the entropy calculation. So I needed to parallelize that as well. But instead of dividing it up among frames, because again, the entropy calculation each frame depends on the other frame, I divided the grid voxels among processes. And by doing that, I was able to then get the total time down to, again, on the order of minutes, and the energy now taking up the most time. And it scales pretty well, you can see on the right. So that was a fun project. And so if anyone out there is using GIST, then be aware that it is MPI parallelized. Here's something that doesn't have to do with parallelism at all, but I think is a cool thing that people should probably be aware about. So I don't know if anyone's had to do any diffusion calculations, right? But typically the way that it's done from molecular dynamic simulations is you calculate your mean square displacement versus time, and the slope of that line is related to the diffusion coefficient. And for systems where your box size doesn't change, there's a heuristic unwrapping scheme that gets rid of imaging, right? And imaging is just, when your particle goes out of the box on one side, it comes into the box on the other side. You have to get rid of that effect if you want to do a calculation so you don't get these unphysical jumps. But it turns out that if you're running a constant pressure simulation where your box size can change, that messes up this simple heuristic scheme, right? It doesn't take into account the box size change. And so there was an initial effort to combat this called the toroidal view preserving scheme from Gerhard Hummer and his coworkers. But one thing that we noticed when I was trying to fix this with CPPTRAJ is that charm didn't have when Charm was doing this unwrapping, it didn't have this problem. And so, oh, I didn't even illustrate the problem. So this top line on the graph on the right, this is what happens if you use the heuristic unwrapping scheme on an NPT trajectory. Eventually, these errors just accumulate, and your mean square displacement line looks like crazy. Charm, on the other hand, didn't have this problem, and I figured out it was because they do their unwrapping in fractional space. So, right, what's fractional space? That's just when you convert your box, whatever size it is, into your unit cell vectors are just all one, and they're all orthogonal. So essentially, you just, no matter what your box looks like, you convert it to a cube. And in that case, the unit cell vectors never change size, and so you never have this unwrapping problem. So it turns out if you just do the unwrapping in fractional space, this is fine. Even more, if you project back onto the average unit cell vectors, um, you can reduce the noise even further. So here on the right, again, I've got at the top, I've got the maroon line, and that's the one that accumulates all the errors. And then I've got the toroidal view preserving scheme, which again restores the nice behavior, but it actually is not calculation wise, it takes a bit to do. And then below it, NVT, that's when the box size doesn't change. That's what it should look like for reference. And then the blue and the black are doing the unwrapping in fractional space. Okay, last thing that I want to mention in terms of new functionality is now you can do energy decomposition with PME with CPPTRAJ. And so that's something that I don't even think you can do in Sander. You can do energy decomposition, but it won't decompose the non-bonded charge part of the calculation. And so you can do full per atom energy decomposition. So what I have here is I have input where I'm doing energy decomposition with PME with a cutoff of 8, and then I'm saving that data. I'm removing everything but the molecules 1 through 3. So again, this is not again, I forgot to mention. This system is F. tularensis Fab I. It's an enoyl reductase in combination with uh, NAD and triclosan. And so what I'm doing is I'm using CPPTRAJ to create a PDB where I'm putting those per atom energies in the B factor column. And so on the right, so you can sort of look at where the hotspots are in this system in terms of the energy. And then down at the bottom, this is sort of the raw output you get. You can get the total energy on the atom and then the contributions from bonds, angles, dihedrals. And again, this is actually parallelized, so you can do this in parallel. But really, it was the being able to decompose the energies with full PME on that's the innovation here. So so yeah, so sort of the last thing that I would like to get into is the use of ChatGPT for creating analysis scripts. And this might be a bit of, old man yelling at a cloud type thing. I mean, I know that, I mean, if you look, machine learning is here, it's here to stay, it's everywhere. If you look at the mentions of, machine learning and ACS publications, I mean, and this graph only goes up to about 2020. So you can just imagine that it's just everywhere now. Um, One of the things that I think is that I love about chatbots is that it will hallucinate, right? Chatbots are like, they're trained to give answers, and sometimes they'll give answers at all costs. So two of my favorite examples of this, and you can just pull this off of Wikipedia. One is that if you ask for proof that dinosaurs built a civilization, um, ChatGPT claimed that there were fossil remains of dinosaur tools and stated that some species of dinosaurs developed primitive forms of art. And that's kind of funny to picture. I can't imagine a T-Rex, the tiny T-Rex arms trying to like carve something into a rock. Um, and then the other one that was really funny was somebody convinced ChatGPT that churros could be used to do home surgery and found that the flavor has a calming effect on patients. And so chatbots are cool, but like they do have a tendency to hallucinate. So we recently we got access to ChatGPT 5 at the NIH, and so I figured, well, okay, let me ask ChatGPT and ChatGPT 5 like to come up with some analysis scripts because this is something that actually I've seen students try and do, and some you can tell sometimes when the script has come out of a chatbot because there are some things that are a little strange. So first I asked ChatGPT to generate um, input for solute to solvent hydrogen bonds and calculate the average lifetime. It's just this is something that's been around for a while. Figured it's fairly simple. So ChatGPT was like, sure. And like started out okay, load the topology, trajectory, good. Gets to the HBON command, okay, out. And then just starts hallucinating keywords. Solvent, that's not a keyword. Solute, that's not a keyword. Lifetime, that's a separate analysis. And so ChatGPT kind of just went off into la la land. Now, ChatGPT 5 thought for 30 seconds, came up with some input. What does it look like? I gotta say, I was pretty impressed. And one thing I was really impressed with is it's all annotated. So it's all explaining what it's doing. And like this way, maybe if it makes a mistake, you at least have a basis to figure out. But ChatGPT 5, it's got the right keywords. It's saving the time series data. It's got the lifetime analysis after the run. This will work. So this was good. I was like, okay, fairly encouraged. So I tried to ask, I tried to ask it to do something different. And that is something that probably, tons of people want to do, principal component analysis. It's based, a lot of people, that's a very common analysis to do. Um, I asked it to perform principal component analysis on all heavy solute atoms of molecule one. And again, ChatGPT, very eager, came up with this. So, okay, it's not bad. It's assuming molecule one is residue one, so it's not familiar with the molecule selection syntax. So okay, that's something you could fix, right? Another thing that it does that's a little strange is it has these two RMS alignments. So the second one will just cancel out the first one. So I'm not sure where that came from. And then here's here's some bad input. This vex. Vex is supposed to be the number of eigenvectors you want, not the file. And so this, I'd give it maybe like a B minus. This is something that if you're familiar with the commands, you could probably fix it. Chat GPT 5. This thought for a little longer, a minute and a half this time. Now it came up with a nicely annotated script. Again, only one mistake, and that is that it does a run analysis before it has the matrix diagonalization to calculate the eigenvectors. This is something that you could fix, but like it loads all the coordinates into memory. It like does the fitting. It calculates, the covariance matrix. So this is okay. Not as good as the H bond one, but like still, if you kind of know a little bit of what you're doing, this would be okay. And then I tried to give it a sort of a real world example, a real question that someone might have. So actually, my old graduate advisor, Carlos Simmerling, asked me a couple of years ago. He wanted something in CPPTRAJ that would help him construct, all atoms within some distance cutoff and then save all those associated distances. And I thought about it for a bit. And there's a new for loop structure. I mean, new. It's been in now for years, but you can, set a reference structure and then just loop over atoms. So anyone who's used CPPTRAJ, the distance selection syntax is a little crazy, but essentially this just means, atoms within 3 angstroms of, you know, residue 4 atom n, right? And so this did what he wanted, and then after I do this loop, I'm writing all that out to, a file. Fine, done. Carlos was happy. So I figured I would pose the same question to ChatGPT. So ChatGPT, again, very eager, but this time just like way off in La La Land. Like first reference, like this won't work. References for loading in a reference structure. And then it came up with a command. It invented a command called radial distances. And so that's just not gonna, I mean, I guess if someone wants to write it, sure, fine, but this won't work at all. So now ChatGPT 5 has been consistently better, except this time it's not. So again, first of all, this mask command here is going to strip down, I think, the system to just this mask. Or, well, no, it just, this doesn't do anything. This essentially will write a file. It's used to actually test like distance select. So like this command is not going to have an effect. It's not bad, but it also doesn't help. And then it uses radial, which again, that's the radial distribution function. That's not going to give you individual distances. To be fair, ChatGPT 5 kind of like realizes that and gave a little caveat and tried to use the closest command to output neighbor distances. This is almost clever, except closest is for molecules. So like if it were molecules, that'd be fine, but for individual atoms, this just won't work. And also that keyword is wrong. It's closest out. So this time ChatGPT 5 couldn't come up with it. So I was like, well, maybe I wasn't clear enough. And I told ChatGPT 5, use a for loop. I told it to explicitly use a for loop. And then it came back and told me that CPPTRAJ doesn't have a C-style for loop, which I guess, okay, to be fair, I guess it's not exactly a C style, but it's pretty close to C style. It's got colons in there, semicolons in there. Um, anyway, it tried and it just made a for, so it invented the for syntax, and it's looping over frames and not atoms, and it's again trying to use closest. So ChatGPT 5 this time, I couldn't save it. I don't know. Maybe someone more clever than me could come up with like a better prompt to generate the input. I think it's because the for loop structure in CPPTRAJ probably isn't as widely used as some of these other commands. And so the training data is limited, and that really limits what ChatGPT can come up with. So um, I think that chatbots have become genuinely impressive. The just getting that H-bond thing right, that was really nice. And providing all those helpful notes, I think could be good. But there's still a tendency to just hallucinate, to just invent these commands and keywords, particularly in the basic version. ChatGPT 5 does not care. We'll just straight up lie to you and we'll do it like really, really happy. So you have to be aware of that. So it might be best used as a starting point, but I still think that looking at the manual or like reaching out to the mailing list, reaching out to your coworkers for like stuff that works. Maybe if you have a script that you know works and you feed it into ChatGPT and say, hey, modify it to do such and such, that might be a better use. But yeah, it's kind of wild how well they how close they come. It makes me wonder a couple of years what they're going to look like. Um, all right, so I was asked to provide a little advice. I don't know if this is the kind of advice people are looking for. If not, then feel free to tune out at this point. But I I figured I'd give a few pointers on anyone out there who wants to develop scientific software, some of the stuff that I've learned. This is maybe it's obvious, but your software must be usable. If your users have to jump through sometimes even one hoop to use your software, they're not going to do it. If they have to jump through multiple hoops, forget it, right? If they have to install this and this and this and this and like eventually they'll just give up. Um, when I wrote CPPTRAJ, I wrote it to be fully backwards compatible with ptraj for that reason, except for the hbond command, because the hbond command originally in ptraj was no good. Sorry, Tom. Um, If you know, you know. Um,
Mhm.
and then also one of the things that Dave Case said to me early on that stuck with me in terms of scientific software development is to adhere to the principle of least astonishment or least surprise. And that's basically like if a user expects something to work a certain way, it should work that way. You shouldn't give it a, you shouldn't have like a keyword that is, say like cutoff and but it means stuff outside the cutoff, right? Usually you think of cutoff as within. So stuff like that. Um, and that really leads into the next thing is to solicit user feedback. And that's something that I try to do, especially with CPPTRAJ, because when I'm developing stuff and I'm coming up with commands, it all makes sense to me. But like, you know, I'm my wife, who's also a scientist, will use it and be like, I like this makes no sense. And so the rubber hits the road moment is when like users actually try to use it and take that feedback and make your program better. If something's confusing, change it and document your code inside it out. That means document inside the code, like in the source files themselves, and then also like write a manual. And I've heard people say this, and I will push back against this forever and ever. Good code is not self-documenting. You may think stuff makes sense, but like someone coming in fresh will not. So like write write out comments. I like to use Doxygen, whatever floats your boat. And write test cases for everything because when you change something, if there's, if you break if some functionality, people are going to be mad at you. And then just some general career advice. You really have to love science. This can be really tough. Experiments don't work out. The hours sometimes are really crazy. Like funding is tough. You really have to love science. It's a lot of fun. Um, but so you want to find an area or topic that you're passionate about. Weirdly, for me, it was like data analysis, like trajectory analysis, but like might be a system that you like. Whatever. Slumps will come and go. You'll have times when like your projects don't work. And like I said, the first two papers that I tried to write were just outright rejected and like I saved some of the comments and they were mean. It was basically like this person like has no idea what they're doing. I was like, yeah, I'm a third year grad student. What do you want, man? But it will pass. Um, this one really apply for grants and fellowships. Actually, the UGA website, the graduate website, has a really cool page where they have a lot of funding opportunities. If you get like a grant, like an ACS grant, and if you come with your own money, you're going to have a lot more options about where you want to go next for like your postdoc or something. And then I really should have emphasized this more and like bold-faced it in big text. Make time for mental health, especially nowadays. Make time for mental health. It's like it's a really stressful time. I mean, grad school is stressful no matter what. This is a particularly stressful time, and I really feel for everyone who's like looking to transition to postdoc or looking for academic jobs. Make time for your mental health, whatever form that takes. And then I think the NIH summer internship program should be back next year. There should be info posted in mid-October. Um, in general, the NIH is a really fun place to be. I hope it continues to be. I'm really hope. Um, so yeah, I think that's it. I just want to acknowledge Bernie Brooks, who's the guy who pays me and keeps me employed, and I like that. I did a lot of this work on the Lobos cluster. I'd like to thank Rajan again for inviting me, and I'd like to thank all of you for listening, and I'll take whatever questions.
Thank you so much. Yeah, time for questions. Olivia, you wanna go ahead?
Sure, I'll ask a question. Thanks so much for this talk. I don't actually really know anything about molecular dynamics, so it was cool to, I liked all the different things that you put in so everybody could find something. to get out of it. And I also thanks for working for the NIH and being a public servant, and thanks for also sharing things like papers getting rejected and stuff like that. I think that's so important for people who are like further along in their career to share, because I feel like a lot of people just share their successes, and then like you could feel kind of bad about yourself if you're getting like you're in the rejection era. So the questions that I have for you are, so I know that now we're kind of in an era where like computing is like really being pushed, especially like in the government and stuff like that. Do you see the NIH like investing more in computing in the future? And do you know anything, just any information you might have on that? Um, that was my first question.
Okay, so it's a bit difficult to answer, and so as everyone probably on this call knows, it's been a challenging couple of months at the federal government. There's a lot that's been changing from day to day. I tell you, it was an interesting time to become the head of an HPC research and have to deal with budgeting and have to deal with like I mean, just literally the guidance would change from week to week. Um, I do know that there is an interest and a push, particularly in the machine learning direction. I mean, it's everywhere nowadays. In terms of actual funding, we're sort of being so everyone also probably knows we may be heading for a shutdown next week over budget stuff. And there have been budgets proposed by people who shall not be named that have as much as a 40% cut to the NIH budget. And so it makes it really, really tough to plan on types of investment. So I think that there is an interest in investing in compute resources, particularly for machine learning AI applications, but just general computing. I mean, the BioWolf cluster is a big cluster at the NIH, and I think they're also interested in funding um, extramural computing as well. But so the desire is there. Whether the means will be there, I mean, a lot of us are just sort of hanging on for dear life. So I know that's kind of a non-answer, and I'm sorry about that, but that's sort of where we're at these days, unfortunately.
Hey Nathan, do you want to go next?
Sure thing. Can you hear me?
Yep.
Yes.
Awesome, awesome, awesome. So I was just gonna ask a question of like, how is your opinion on like modern like I guess structure prediction on the MD space? Because like part of me thinks that like I think it's been like a very good thing now that we see a lot of like structures that have never been resolved before that we can like do MD as like or at least use the predicted structures as like starting points. Do you know of any cons that I'm not thinking of? Because I've been
So
thinking
you're
about this
talking
for a
about
while.
like AlphaFold,
Yep, AlphaFold Tier Zeta,
right?
more of like the fusion flow matchy. Like,
So it is great. I think that it's been a great effort and pretty successful in some respects. I do hate when it's framed as like the protein folding problem has been solved because that's not protein folding, right? Structure prediction and
I agree.
protein folding are two separate areas. But I think that again, some of these efforts suffer from lack of training data. So if you look at AlphaFold predictivity for like protein systems, it's pretty great. Comes up with a lot of good stuff. If you look at it for things like nucleic acids and like RNA, there are some problems. So like there's some it's not completely bad, but there's a recent paper that actually just came out from Alex Gershayev and Christina Berganzo showing that specifically for nucleic acid systems, that there are some real issues with the structures generated by AlphaFold. And I think, and it makes sense, um, that it just comes down to there's less data for nucleic acid systems compared to the wealth of data there is for protein systems. So I think that it's great, and I think that it's an ongoing effort. Um, and I do think that it is important that we continue to verify as much as we can, just because, there are still some gaps in the knowledge. But it's really amazing just some of the stuff that comes out. It really is cool.
Well, thank you so much for that answer. I 100% agree with you. I think if we can't validate it, it's hard to say it's right. But yeah, no, I 100% agree as a data scientist myself.
Cool.
I see Georgia. Do you want to go?
Hi, can you hear me?
Yes.
Yep.
Thank you, Dr. Rowe, for this presentation. And also, I agree with Olivia that it was so nice to hear from someone on NIH. I have a question. I think I'm not sure if it makes sense a lot, but you were saying that you have been in this space for 24 years, like you showed it, like started in 2001. Um, and I think the as you were saying in your graphs, like the way CPUs and GPUs have evolved has quite literally changed how much data you can generate and process. So has there been any problems that you like thought of 20 years ago and then you have satisfactorily solved probably sometime recently that made you feel like, yeah, that is a progress that I didn't imagine I could do it probably 20 years ago, but it is possible now.
It's fun. So I think I'm guessing you're talking more about like a sort of a research problem than like a software development thing, right?
Yeah,
Because yeah,
any of it makes sense.
so there are things that I've thought about going back to. I just have not had the time because basically like I have no time, but there was a really cool system that I studied when I was in grad school that was like this iron transporter protein that was this giant, it was like Ton B or something. Like I don't even remember the exact name, but it was cool. It had this like little protein cap. It was embedded in a membrane and like it facilitated iron transport. And like I remember proposing this and Carlos being like, this is nonsense. It's like it's embedded in this lipid membrane. We don't even have parameters for this. Like this would just take forever. And I really wonder, like now with the advances in like speed and parameter development as well. I mean, the parameters we have now, the force fields are great. Um, I would love to, yeah, if I had world enough in time, yeah, there's definitely stuff that we can do now that I couldn't think about even attempting before, mostly with those bigger like membrane type systems.
Thank you for the answer. It really feels good that you can imagine of things like that.
Mhm.
Yeah.
Next, MHM, you want to go?
Sure. Thank you so much. It was a great talk. I loved every part of it. I have two questions. One is like you explained the dice issues, like how it is tough to reach to a convergence. For like molecular dynamics, especially with the glycan or carbohydrates, we see a lot of different behavior. Like each MD will show different types of movement for the glycan especially. So I was wondering if you have any thought on that, like how long we might have to run for the simulation. The one I'm talking about is like 1 or 2 microsecond. Even within those 1 or 2 microsecond, we are not seeing same behavior for those glycan chain as those are very flexible. And second question is, how do you compare CPPTRAJ with the other tools like MDTRAJ? Or do you know any other alternatives that are very good with for this purpose, like for say carbohydrate?
Well, so I don't, well, so maybe I'll go in reverse order. I don't know, I don't often compare against other analysis tools, and it's not because I'm not interested. I just don't, I tend not to use them, but I do know they get a lot of use. So like MD-TRAJ, MD analysis, um, loose, PyLoose. A lot of them are Python based, and I came up in the era of Python where like Python's gotten a little more stable, but like there was an era, especially in Python 2, where like packages were like changing and breaking constantly, and it drove me nuts. Like I write a script, I want it to like work 10 years from now. I don't want to have to like redo it. Um, so
Mhm.
in terms of so I mean I don't know in terms of like carbohydrate specific analysis if one package is better than the other. If there's functionality that should be in CPPTRAJ, it's something that I would encourage people to put in there. I think that the big advantage, and I don't know if this is true anymore because I haven't had a chance to keep up on it. The big advantage with CPPTRAJ is if you need to do highly parallelized analysis, I don't think the other packages do that quite as well, although I could be wrong about that. In terms of the convergence issue, again, you know, MD on biological systems, much more complicated, many more degrees of freedom, and it really depends on what energy barriers you have, right? It's convergence is going to be slowed down if there are high energy barriers that you're not crossing frequently. And so, like, a good example of this is if you have a system where, like, proline isomerization is like a cis trans isomerization is like a big thing, right? Because that will you're going to converge at the rate of like whatever your slowest conformational change is. And so it really depends. And there are some things for which like replica exchange won't get you much. If you're looking to do a full folding landscape, replica exchange might not be the best because folding's not necessarily accelerated by temperature. You might have to do a different, modify your Hamiltonian in different ways or use a different enhanced sampling method. But like, it really is sort of system specific. And the only way to start to evaluate convergence is running independent multiple simulations from different points on your free energy landscape. And that's only at that point. And then you do things like you can do combined clustering analysis, combined principal component analysis, just comparing, RMSD, first half, second half. I mean, just having independent simulations is really the only way to properly evaluate convergence.
Okay, great. Thank you so much.
Yeah. We are about at the end of the seminar, but I have one question. So for the replica actions MD that you were saying, uh, they are all, the serial processing, calculations, right? So I was wondering, do you see any potential in parallelizing the replica actions MD?
Oh, the analysis?
The MD itself, like do you
Oh,
see any potential in
well,
the
replica exchange is parallelized, or in fact, you can't, at least in Amber, you can't run replica exchange without using parallelization. Right. So
with
each
Mhm.
replica essentially gets assigned to a different group, and each group can have multiple nodes. Yeah, I mean that's how we were running replica exchange on Blue Waters. Each GPU was getting its own replica. Yeah,
Okay.
so really, I mean that's the advantage. Now I know there are attempts to put multiple simulations on a single GPU, and you can
Uh-huh.
do that. It's escaping me right now. There's like a native partitioning you can do for like recent GPUs where you can split them into like seven. And
Okay.
but yeah, like replica exchange sort of from the get-go was envisioned as being a parallel method.
I see. I see. Okay. I think we have to call it the end of the seminar. And again, thank you, Dr. Ru, for presenting in the seminar today.
Yeah, that was fun. Thanks for the invite.
All right. See you all.
Take
Have a
care.
good day.
You too.